My name is Mark Vloosdrecht, and this is the second lecture in the module on elemental cycling by microorganisms. In this session, we will focus on the anaerobic conversion of organic carbon, transformations that occur in the absence of oxygen. In the previous lecture, we discussed aerobic processes where microorganisms ensure that all organic carbon produced on Earth is ultimately converted back into carbon dioxide. In some cases, this occurs indirectly through anaerobic conversions that produce methane. This methane can then escape into the oxygen-containing areas, where it is oxidized to carbon dioxide, closing the carbon cycle. In this lecture, we will concentrate on anaerobic microbial conversions. These microbial processes are more complex than aerobic ones. For example, under aerobic conditions, a sugar molecule can be completely oxidized to carbon dioxide by one single organism. Under anaerobic conditions, however, this same sugar molecule must pass through multiple sequential steps involving fermentation and methanogenesis and a range of microorganisms. The methane produced in anaerobic environments can diffuse into aerobic environments, where it's finally oxidized to carbon dioxide, completing the carbon cycle. Anaerobic processes can be broadly divided into two categories: the anaerobic respiration and fermentation. In anaerobic respiration, microorganisms grow by transferring electrons from organic substrates to alternative electron acceptors, other than oxygen. Examples include nitrate, sulfate, ferric iron, and manganese oxides. In principle, any possible redox reaction can serve as an energy source for microbial growth. The biochemical mechanisms are similar to those in aerobic respiration. Electrons are first captured by intercellular carriers such as NADH and then transferred to an electron acceptor, not oxygen in this case, but one of these other compounds, generating energy in the form of ATP. When no suitable external electron acceptor is available, fermentative processes take over. In these pathways, part of the substrate is oxidized to CO2, while another part is reduced to products such as ethanol, acetate, or finally methane. Alternative electron acceptors generally yield less energy than oxygen, which means that anaerobic microorganisms typically have lower growth rates and lower biomass yields compared to aerobic organisms. Anaerobic respiration provides unique opportunities in environmental biotechnology, since a wide range of redox reactions can serve as energy sources. These microbes can degrade compounds that are difficult to treat aerobically. For instance, tetrachloroethylene can act as an electron acceptor and be sequentially reduced to trichloroethylene and hydrochloric acid. Through this process, bacteria can use such pollutants in their energy metabolism, ultimately transforming highly chlorinated compounds into non-chlorinated compounds, less toxic forms, which can be easily removed under aerobic conditions. This makes anaerobic respiration particularly valuable for the bioremediation of halogenated pollutants, including chlorinated, brominated, and certain fluorinated compounds. Fermentation occurs when no external electron acceptor is available. In fermentation, one portion of the organic carbon is converted to carbon dioxide, while the remaining carbon is reduced to a more reduced compound. Overall, the process must remain redox balanced, meaning the total number of electrons in the system remains constant. This can be visualized by examining the oxidation states or the valencies of the atoms in substrate and of the products. Common examples include the conversion of glucose to CO2 and ethanol, the well-known alcoholic fermentation used in brewing, or the conversion of acetate to CO2 and methane in methanogenic environments. Fermentations generate very limited energy for growth because most of the substrate's energy remains in the products. Ethanol and methanol are energy-rich substrates or products. Consequently, these microorganisms grow slowly and yield little biomass, require longer retention times in biotechnological systems. The final stage of anaerobic carbon conversion is the formation of methane. Remarkably, there is no single organism capable of converting simple substrates like glucose directly into methane. Instead, methane production from glucose requires a consortium of microorganisms working together in a syntrophic ecosystem. Hydrolytic and fermentative bacteria first break down complex polymers such as proteins and polysaccharides into monomers, amino acids and sugars. These monomers are then further oxidized or fermented into fatty acids and alcohols. Syntrophic bacteria can convert these intermediates into acetate, hydrogen and carbon dioxide. Finally, highly specialized methanogenic archaea, as they are called, convert these compounds, mainly acetate and hydrogen, into methane. Each group in this food web depends on the others. Methanogens are extremely specialized, often able to use only one substrate type. Likely this is related to the fact that the energy yield of their conversions is extremely low. Their specialization allows them to survive and function efficiently in strongly energy-limited conditions. In environmental biotechnology, anaerobic processes are particularly useful for treating high-strength wastewater, as in industrial wastewater, and solid organic waste, where large amounts of organic material need to be removed. As with aerobic treatment, the goal is to remove organic carbon before wastewater is discharged into natural waters, preventing oxygen depletion and protecting aquatic life. A major additional benefit of anaerobic treatment is the production of biogas, primarily methane, which can be captured and used as a renewable energy source, for instance to power engines or generate electricity. Anaerobic processes are effective in removing organic pollutants, but they have some limitations. The low growth rate and low biomass yields require longer retention times and larger reactors. The processes are generally more sensitive to disturbances than aerobic systems due to the low availability of energy for growth. Nevertheless, anaerobic systems are widely applied, especially for biomass production and energy recovery from waste. However, because not all compounds are completely degraded under anaerobic conditions, these systems usually require a subsequent aerobic post-treatment step to achieve a good effluent quality and fully clean water. To summarize, anaerobic conversions play a vital role in the global carbon cycle, in industrial wastewater treatment especially. They offer major advantages. They efficiently remove organic carbon. They produce methane or biogas, supplying renewable energy to industries or society at large. Unlike aerobic conversion, where a single organism can complete the process to CO2, anaerobic conversion relies on a complex microbial consortia working in close cooperation. Through these interactions, nature and biotechnology achieve efficient carbon recycling even in the absence of oxygen.